News release, Monday, March 27, 2023. National Institute of Health-funded study confirms white blood cell subtype as HIV reservoir, suggests new target for cure efforts. A subset of white blood cells, known as myeloid cells, can harbor HIV in people who have been virally suppressed for years on antiretroviral therapy, according to findings from a small study supported by the National Institutes of Health. In the study, researchers used a new quantitative method to show that HIV in specific myeloid cells, short-lived monocytes, and longer-lived monocyte-derived macrophages can be reactivated and infect new cells. The findings, published in Nature Microbiology, suggest that myeloid cells contribute to a long-lived HIV reservoir, making these cells an important but overlooked target in efforts to eradicate HIV. Our findings challenge the prevailing narrative that monocytes are too short-lived to be important in cure efforts, said study author Rebecca Veen-Hues, Ph.D., an assistant professor of molecular and comparative pathobiology and of neurology at Johns Hopkins University School of Medicine, Baltimore. Yes, the cells are short-lived, but our follow-up data show that HIV can persist in monocytes over several years in people who are virally suppressed. The fact that we can detect HIV in these cells over such a long period suggests something is keeping the myeloid reservoir going. The study, led by Veen-Hues and colleagues at Johns Hopkins University School of Medicine, was supported by the National Institute of Mental Health, the National Institute of Allergy and Infectious Diseases, and the National Institute on Drug Abuse, all part of National Institute of Health. Antiretroviral medications are effective in treating HIV because they prevent the virus from infecting new cells and multiplying. However, HIV may still exist in cells that are in a resting or latent state, creating an HIV reservoir. CD4 T cells, a type of white blood cell, are the most well-studied HIV reservoir. Identifying HIV reservoirs is critical to cure efforts, as latent HIV can be reactivated if people stop taking antiretroviral medications. Monocytes are immune cells that circulate in the blood for about 3 days before traveling to tissue in various parts of the body, including the brain, where they can mature into macrophages. To date, it has not been clear whether latent HIV in these cells can become active again and infect other cells. What's really important in the long run is understanding how monocytes contribute to the tissue macrophage reservoir, explained Janice Clements, Ph.D., senior author on the study and professor of molecular and comparative pathobiology at Johns Hopkins University School of Medicine. If monocytes can carry virus to the brain or lung or another part of the body and infect resident macrophages that are self-renewing and live almost indefinitely, that's a real problem. In the study, Veen-Hues, Clements, and colleagues first measured HIV DNA in myeloid cells in a sample of 30 participants with HIV, all of whom were virally suppressed and had been on antiretroviral therapy for at least 5 years. They found detectable levels of HIV genetic material in monocytes and macrophages, though the levels were much lower than those observed in CD4 T cells. In some participants, the HIV genetic material found in monocytes was intact, which suggests it may be capable of infecting other cells if reactivated. They then used the new quantitative method they developed to directly measure viral spread from HIV found in myeloid cells. The researchers isolated monocytes from blood samples taken from 10 participants and nurtured the monocytes in cultures that contained antiretroviral drugs to replicate the participants' baseline physical state. After the monocytes differentiated into macrophages, the researchers introduced an immune activating agent and then added fresh white blood cells to allow for the virus to spread to new cells. The researchers collected samples from the cell cultures several times over the next 12 days. They included checkpoints throughout the process to ensure that infected CD4 T cells did not interfere with their measurements. The results showed that cultures from 5 of the 10 participants had detectable HIV genetic material in monocyte-derived macrophages that could be reactivated to infect other cells and produce more virus. The participants, who had these reactivatable reservoirs of HIV in monocyte-derived macrophages, had higher overall levels of HIV DNA material. Follow-up data from 3 participants showed that this reservoir can be long-lived, harboring latent HIV for months to several years. These reservoirs were stable and could be reactivated over time, indicating that monocyte-derived macrophages could contribute to viral rebound if antiretroviral treatment is disrupted. The researchers note that this study is small, and larger studies with more diverse participant pools will be essential to accurately estimate the proportion of people who have latent HIV in myeloid cells. Investigating the mechanisms that replenish the monocyte reservoir over time is a critical next step in this research. These findings underscore the importance of broadening the scope of HIV cure efforts, said Joshua Gordon, MD, Ph.D., Director of the National Institute of Mental Health, shifting away from a sole focus on CD4 T cells to thinking about CD4 T cells and myeloid cells together in context will propel the field toward more promising strategies for eradicating HIV. Grants M127,981, M113,512-3, Zone MH75,673, Ione 131,306, Ione 127,142, Ione 4,189, D 50,529. About the National Institute of Mental Health NIMH. The mission of NIMH is to transform the understanding and treatment of mental illnesses through basic and clinical research, paving the way for prevention, recovery, and cure. For more information, visit the NIMH website. About the National Institutes of Health. National Institute of Health. National Institute of Health, the nation's medical research agency, includes 27 institutes and centers and is a component of the U.S. Department of Health and Human Services. National Institute of Health is the primary federal agency conducting and supporting basic, clinical, and translational medical research and is investigating the causes, treatments, and cures for both common and rare diseases. For more information about National Institute of Health and its programs, visit www.nagol. National Institute of Health Turning Discovery into Health Registered. Veen Hughes R. T. Devru and Costa and She Ferreira and Ratliff J. Politz L. Shirk and Rubin H. Blankson and Gamma L. and Clements 82023. Monocyte-derived macrophages contain long-lived latent HIV reservoirs. Nature Microbiology. https://doi.org/10.1038/S40 1560 4-23-1349-3